wherever we can find it again. Okay, cool. All right, everybody. Hi, I'm Terri Conneran, and I am just thrilled tonight to be able to introduce to you Dr. Hossein Borghaei from Fox Chase in Philadelphia. He is, oh my gosh, what? You're like almost a rock star up there.
Oh.
Okay, I don't believe that's part of your official title, but like chief of Thoracic. Uh-oh, we're losing you. I lost you. I lost somebody. All of a sudden, I've got somebody else's screen up here. But you're like chief of, let's see, somebody's viewing their screen. Hello? Help me. This will stop sharing other screens. Yes. Hang on a minute. Let's take care of our technical difficulties. And okay, I think I think we are back. Correct?
Yes, we're good.
Okay. Well, that was a relief. Okay, well, welcome to the world of Zoom. And you're just chief of thoracic oncology at Fox Chase, and also just accepted and agreed to be on our board of the KRAS Kickers. And we're super excited to have you because we know that you're out there and you're working hard to kick cancer's KRAS. And so I am just totally thrilled to have you not only there, but also our first expert in our expert series. And so thank you for joining us tonight.
It is my absolute pleasure to be here with all of you today. I hope that we can have a nice, friendly conversation. So I'll do my best to answer as many questions as I can, and I might have some questions for you guys. And Terri, thank you so much for putting all of this together and for inviting me.
Okay, well, great. Okay, well, let's just kind of start out super general, and then we'll just kind of go into it. And so how would you describe what is a KRAS? What is KRAS? What is a KRAS biomarker?
So in order to answer that question, I think we have to define a couple of different things. So terminology. So doctors talk in jargons a lot, and I'm trying to avoid that. So let's define what we're talking about. So we have identified within the DNA of all of the cancers that we examine, areas of abnormality within the DNA that we call simply mutations. So that's one way of talking about some of the changes that we see within the DNA of a tumor cell. So how do we do that? We do a biopsy, we isolate the tumor, we take that tumor to the lab. We are able to identify and isolate individual cells out of that tumor that comes to the lab. And then we extract the DNA from the tumor, and we do something called sequencing. Sequencing is just a matter of finding what is the alphabet for that particular DNA in that particular tumor. The alphabet of the DNA is what makes all of us, if you believe in the world of biology. So it is the alphabet that gets translated in all the proteins that eventually make us what we are. And that's actually true of all the organisms in on this planet. A mutation happens when the alphabet is not translated or not copied exactly as it is intended to be. If you imagine that we have like a 4-letter alphabet for the DNA, sometimes instead of putting an A where it's supposed to be, something goes wrong and a mistake is made, and instead of an A C is put in or a T is put in. And that causes what we call a mutation. And these mutations can have significant implications down the road because one change could actually stop the accurate translation of the alphabet in that particular DNA of that particular cell. And it's exactly these kinds of mistakes, mutations, that can lead to formation of tumors. Now, how does that happen?
Well, hang on one second though. Okay, so let me see if I've got this right. So you're saying this would be like a computer code. right? And you know how we're all out here on our Zooms and our emails or whatever, and we're copying and pasting, but I copy and paste it, but it misspells like one word.
Yeah.
And then I copy and paste it there. And then it just keeps taking it and replicating, keep duplicating it so that it's still wrong further on down the line. Is that what you're saying?
Basically, but it's not just even a whole word. It's just sometimes one letter can change the meaning of the entire word, and then that word is inaccurately translated and goes hand down, down, down. And we see this in really old texts. In many religious texts and many non-religious texts and philosophical texts. If you go back, hundreds of years ago, people used to copy these things by hand, and they would make a mistake, and that mistake got carried all the way down to us until the scholars were able to go back and find a newer copy and say, well, this doesn't make any sense. So a mutation is simply that. It's a mistake in copying and writing the correct alphabet or code down. And that's what can get us into trouble. Now, our cells are actually extremely smart. We have this machinery that can actually recognize a mistake and correct it. So as our cells try to divide, there is actually a very complicated machinery that says, hey, it's supposed to be a C here and it's a T, and I'm going to correct that. So a lot of our bodies, it's amazing how we can correct these mistakes. But once in a while, it just gets out of control, and you cannot correct it.
So like so in that sense, how is like the KRAS G12C? How would that fall into the, or D or S or whatever? How does that fall into the explanation there?
Well, the G12C is a very specific mutation under the whole KRAS umbrella. So KRAS is the most common mutation we find in non-small cell lung cancer. But what we have recognized over the last decade or so is that KRAS is actually a number of different mutations, meaning a number of different changes, single letter changes in this big gene that can get translated into protein. So the G12C, the one that we're sitting right now, happens to be one of the more common subtypes of KRAS. It's still KRAS, but it's slightly different. And the reason we're so interested in is because we have a couple of drugs, and we're going to talk about that a little bit. So the difference here is that we think these different letter changes within the KRAS actually can lead to mutations that make a cancer cell behave either more aggressively or sometimes not really all that different from a KRAS from a tumor that's not KRAS mutated, meaning that these single letter changes can actually change the characteristics or the behavior of the particular tumor. And this goes all the way to response to chemotherapy. There's some old data that one particular form of KRAS, let's say G12C, might respond better to chemo, but G12T would not respond to chemo. So there is that level of granular data for different subtypes of KRAS depending on these small letter changes. The consequence of a mutation in the KRAS gene is that genes are what we call translated into proteins. So you get the gene, you make a protein off of it, and the protein basically directs the cell to do what it is supposed to do. So a mutated KRAS leads to a mutated protein. And in this case, the mutated protein instructs the cells to continuously grow, never stop. And usually cells have a shut off. There's an auxiliary that says, okay, enough, you got to change, you got to stop. Well, when you have a mutation, that doesn't really happen, and the cell grows uncontrollably. And that by definition is cancer. It's an uncontrolled growth of a cell as a result of a single mutation that leads to a bad protein that doesn't know when to shut off. And our job is to try to figure out, well, how do you shut that bad protein off, or how do you interact with that mutated gene so that you don't have an awkward looking protein that's constantly on. Does that make sense?
Yeah, so is that kind of when you're saying it gets stuck on and it doesn't want to come off? And that's where these checkpoint inhibitors start falling in, is that right? Where we start stopping them?
Okay, so we got to be careful what we call checkpoint inhibitors, because right now in the world of lung cancer treatment, checkpoint inhibitors are specifically used for immunotherapy checkpoints. But you're right in that in the cell cycle process, there are places where we call them checkpoints simply because it's a point at which the cell has to decide, do I go through it or do I not go through it? So it's a checkpoint, right? Do I have a passport to go through or don't I have a passport and I can't go through the border, right? So it's a checkpoint. But in general, right now, we're using the term checkpoint inhibitors mostly as it applies to immunotherapy, which is a different concept than directly interacting with the mutated gene.
So the inhibitor is the part that's stopping it. But the checkpoint inhibitor, usually when somebody says that, they mean the immunotherapy, not the targeted therapy specific to the KRAS. Is that right?
Yeah, it's just the terminology. That's the way. Now, I got to tell you, what I tell you is from a clinician point of view, from a scientific. So if you talk to a PhD level geneticist or a molecular biologist, they use the terminology a little bit differently. So when you have a speaker, when you're talking to someone, you have to make sure that you're all on the same page. From a clinician point of view right now, the term checkpoint inhibitor usually is associated with the use of immunotherapy drugs. But what we're talking about here are targeted therapy. But if you talk to a scientist, they might change the terminology a little bit. So I think we have to be a little bit careful with that.
That's a really good point, because a lot of times we're out here, we're just drowning and trying to find out what's going on, right? And we don't necessarily know what it is like we're listening for. So that's really valid. Now, and as we have started going down towards like the targeted pathway, right, towards the precision medicine, and we've all heard about, where, and if you're here, you've heard something about KRAS G12C. You may not understand everything about it, and that's cool. But before we even get into possibilities, why has this been so hard? Why has it been so hard to get to this?
That's a little bit more difficult to sort of answer. It's been more difficult to target KRAS because A is not a single entity. As I said, there are multiple subtypes, G12C and D and V and all of that. So it's been difficult to sort of find something that hits all the subtypes of KRAS. And second, because to make drugs that specifically interact with one tiny piece of a gene, you got to be able to get the drug to bind into something, because that's how things work. It's like a lock and key type of a thing. There's a lock. For every lock, there's a key, right? And you have to get the right key to open the right lock. So in designing drugs for these specific mutation, you got to be able to find a groove. You got to be able to find something that the drug that you're trying to make can actually stick to. And obviously, where you want the drug to stick, it has to be someplace that's essential for the functioning of that particular gene, so that you stop it from doing whatever it is that's aberrant and not right? So that has not been easy in the case of KRAS. Again, one, because there are so many different subtypes and they all have a different conformation. It's hard for me to show it with my hand. So if a normal gene is supposed to be like that, and you have a mutation that is like this, and at the same time you have all these other intercalating stuff, you got to find the drug that actually can bind between my thumb and my index finger here. And that's difficult to find drugs that can actually go in there and have the biological function that you actually want. So the KRAS has been, for many years, we were taught when I was a fellow and coming up, you just can't drug KRAS. So if you want to inhibit KRAS, you got to go above it a little bit. So find something that interacts with it, or go below it and find stuff that KRAS has caused and try to stop the pathway that way. And that brings up another concept. For some of you who might have engineering backgrounds, or might have seen like the schema for an electrical outlet, that's the way these genes and receptors work. So with KRAS, something binds to it. There's a signal transduction, as we call it. So the interaction on top of the cell has to get inside the cell, and that happens through these multiple proteins that come together and sort of say, oh, something bound on the surface, now we have to take it and bind where the DNA and all that is, and cause whatever effect that we want to do it. And
all right. I'm going to tell you, my son's an engineer and you're losing me. But okay, so here's the way I get it. Tell me, totally correct me if I'm wrong, okay? How I view a pathway is kind of like a waterfall, okay?
yeah,
And and you're trying to get to a specific crevice coming down that waterfall. Now you may be able to come down it, you may be able to go through it on the side, you may be able to find, I don't know, some way to go up and into it, but you're trying to get to one specific crevice. And these rocks are super slippery and you want to be in one specific spot. And am I anywhere close?
no, I think that's not an inaccurate way of thinking about it at all, because it also tells you, just like a waterfall, there are many ways to get at the bottom. And that's why these cells are sometimes tricky. And that's why sometimes it's hard to target exactly what you want and get the desired effect, because you block the waterfall at one level. Let's say your goal is not to have the waterfall in that particular crevice, to get all of it to the bottom. Let's say that's our goal. Well, the water is going to find another way to get down. And this is what cells do. They find yet another path to bypass your block. And that's where these resistant ideas come up. And that's another topic that I'm sure.
The resistance would just be like dams, right? I mean, so like if you put a big old piece of wood or something up on a waterfall, right? Then that's just kind of the same thing, right? I mean, I like I said, I'm not an engineer, and I don't pretend to be one.
You're on the right track again. So when we have the mutations and we find a targeted therapy for it, right? The cancer usually responds, the tumors shrink, all of that is good. We like to see that. But in every case that we know so far, unfortunately, the cancer cells learn to live with that drug and become resistant to it.
Okay.
The resistant mechanism is the example of you have stopped the water in the waterfall in one place, but the water has to get down and it finds another way. That is the resistant pathway. So in order to find better treatments, then we go and look and say, okay, what is the resistance pathway? And can we have a drug for that? And then we say, now can we use two drugs? One for the KRAS pathway for the G12C, and now one for this resistant pathway that we have identified. Because if we can do that, then maybe either the resistance won't happen or it happens way down the road, which is really what we want to see. So all of these things have implications. I just don't want you to get the feeling that because we have a drug that specifically hits this G12C and the results are really good and promising and everything, the work is done. The work is going to have to go on, because we know these cancer cells are going to find a way to become resistant to the drug. So our job is, what are we going to do about it? How do we identify these resistant mechanisms? And what do we do to treat the resistant mechanisms?
Okay, and so a resistant mechanism is something that it may happen, and it often does, so that we can kind of like work around it, right?
Exactly. Yeah, we have to find a way to do it. Sometimes, we ask our patients to participate in these clinical trials where we actually ask, well, can we do a biopsy when the cancer is getting worse, or if it's not responding to the drug? Because that's when we want to be able to identify what these potential resistant mechanisms are, because without actually having another biopsy, when the tumor is no longer responding to the particular drug, it becomes difficult for us to guess what's going on. So asking our patients to participate in the repeat biopsy sometimes helps us in identifying these potential resistant mechanisms, which then again down the road hopefully will help us figure out, well, is there a better drug or a better drug combination that we can develop? So this is complicated, we don't want to ask for biopsies. We're going to try not to do biopsies as much as possible. The technology with the blood-based assays and all that hasn't gotten to a point where we can answer all of the questions that we want with the simple blood draw, which obviously would be a lot easier. So we understand the difficulties in asking for a repeat biopsy, particularly in lung cancer. But sometimes you really have to do that. And it becomes this collaborative agreement between someone who is on a clinical trial and the investigators, trying to make sure everybody understands why we're doing these and why the biopsies are important and why the biopsy can actually lead to better treatments down the road. So these are all issues that I think we all have to talk about it, and we all have to be aware of it if we want to make progress and we want to make find a better drug or a better way of treating the cancers that we're dealing with. And look, without your participation, without patients coming and going on clinical trials, we wouldn't have had all of these advances. So I think there's a lot of credit that has to go to patients who go on these clinical trials for these drugs. And sometimes, unfortunately, the benefit is not all there for the subject who goes on a trial, but people after them would benefit from all of these investigations and research. And sometimes, like we have in G12C, everybody benefits. We all learn the drugs work, and we have a new promising treatment, and now we're just going to build on it, and we're just going to make it better, and that's the next step in this process.
So the G12C, even though it's not going to be working for the other subtypes, is going to benefit the other ones in what way?
Well, now that we know how to make a drug for G12C, it's not as big of a challenge to find something for the G12D, because the sort of, again, we call it conformation, or the structure of the protein or the shape of the gene and all that might be just a little different. Now that we have the basis as to how to get a drug into G12C, we might be able to, well, not might be, I know that we're able to get and make something for G12D, which is the next common one probably coming up. And then you got to keep in mind, the door now is completely open. So there are people who are developing drugs that hopefully will address the entire KRAS, not just the specific subtypes. I think that research is still sort of preliminary and needs some work, but we're beginning to hear talk about drugs with broader applicability coming for clinical investigation. So we'll see how these drugs work. So every little piece of understanding today about any of this helps us down the road understand the rest of the biology and the rest of the KRAS and all the other stuff that goes along with this.
Okay, and how does that kind of like fall into with like the comutations? Because we hear about them, we see them.
Yeah.
Can you help?
Well, commutation, just as the name suggests, it just means that, well, a cancer cell can have more than just one mutation. So you can have a KRAS mutation, and you can have something called an STK11, just indicates another gene that's altered. Or you can have something called the KEAP1 mutation, which is again another gene that can be altered. So, and sometimes having 2 or 3 mutations within the same tumor can have implications for treatment and response to specific targeted therapies, and what you can do down the road in terms of treatment options and all of that. Well, how does that work? Well, if you have one change here that leads to an error and another change here that leads to another error, let's say the change number 1 make the cancer cells more sensitive to chemo, as an example, but all of a sudden you have change number 2 that reverses that and makes the cancer cells resistant to chemotherapy. So the implication of comutations is that sometimes these other alterations can impact direct therapy, even targeted therapy that you have designed for, let's say, the G12C. And that's probably, and I say probably, what we are seeing with a couple of comutations that we're beginning to see, like the STK11 and KEAP1, which are the names that you probably see or read about if you go search into KRAS and comutations. And that matters because the emerging data, the data is not complete, at least in my view, but the emerging data suggests that some of these comutations can impact the response of the G12C KRAS to the specific drugs and lessen it a little bit. So then we have to figure out, A, is that really true? which means that we have to examine a larger number of tumors with co-mutations, and B, to what extent the co-mutations are impacting the response to targeted G12C drugs. And if that's the case, what are some of the ways that we could potentially use combination of treatments to overcome whatever the problem is? So that's why the co-mutations are becoming important and are something that we are paying attention to. We're paying attention to it in every aspect, in every subtype of lung cancer. These co-mutations have implications for immunotherapy. There's early results suggesting that if you have KRAS plus a KEAP1, immunotherapy might not work quite as well. I mean, it still works, but not as well as if you don't have one. So the implication here would be, well, if I have a patient that has all of these co-mutations that might not make immunotherapy work as well, then I have to come up with a different management style or management option for these particular tumors. Again, these are all a little bit preliminary. I don't think anybody's like changing their clinical practice necessarily, but we are learning about them. We're isolating data. We are sequencing more tumors, and we're hoping that we can understand the biology better so that we can come up with better treatment options down the road.
Is that one reason why it's important for us to get another biomarker testing when we have, say, like a recurrence or a progression?
I cannot underemphasize the importance of having another biopsy, especially when there is these targeted therapies like KRAS G12C, EGFR, ALK. There's a lot of good data that repeat biopsies at progression could potentially identify these other mechanisms that could help us figure out what the best next treatment is in this setting.
Yeah, and as much as we simply just want to be able to take a pill and just one and done, like it's the Tylenol, it doesn't sound like it's going to be quite that simple.
Well, again, again, I'm not going to sugarcoat anything, and I'm being very honest. No, it's not going to be quite that simple. Again, the cancer cells, this is a very dynamic state. We are trained to think about these things as fixed in place, and that's not what our bodies are. Our bodies are in constant motion. There's a huge amount of changes every day that happens, and the cancer is the same because it comes from us, because it's from our own body, right, from our own cells. So because of the dynamic nature of this interaction between a person, the immune system, and the tumor, and all these mutations, things constantly change. And cancer cell finds another pathway, and we're going to come up and try to figure out another way of fighting it and stopping it. So yeah, it is a very dynamic state, and that's the reason, again, we do so many biopsies, and there are so many drugs that are being tested for all sorts of subtypes of lung cancer.
So if your doctor doesn't suggest that you get retested, is it something that you should ask for? I mean, how does somebody who's not your patient go about doing that?
I would say there is nothing wrong with asking your physician, do I need another biopsy, and is the biopsy going to be helpful in this setting? Some of the direct questions are, if I do another biopsy and you find another resistant mechanism, do you have a clinical trial, or can you refer me for a clinical trial someplace close to us? So I think there's nothing wrong with at least raising the question. If your doctor comes and says, well, the CAT scan says that the tumor is a little bit worse, we're going to have to stop this drug. So yeah, it's a perfectly legitimate question. Do I need another biopsy? You're going to do another sequencing, and if you find something, can you refer me for a clinical trial if you don't have any?
How do we ask for that? I mean, it feels, I don't know, awkward. Like it does as a patient. You're like, hey, can you refer me for a clinical trial? I mean, should we feel weird about that?
At the end of the day, you're your own best advocate. The more educated you are, the better you can take partnership in your treatment with your doctor, right? I don't think there's a doctor here who wants to sort of not do what the right thing is for a patient, right? But as physicians, sometimes if we don't have an immediate treatment option for our patients, we sort of feel it like, it's a deficiency on our side, and we sort of want to get to the next treatment so that we can tell the patient we're doing something about this. sometimes the right thing to do is to take a step back and say, okay, where are we? What are the goals? What do we need to accomplish? And the question of whether there is a trial that can be applicable to my tumor is a perfectly reasonable question. I don't think any medical oncologist in this country is going to be upset if you say, is there a trial out there for me? It's the same thing with the repeat testing for a repeat biopsy and molecular testing. You're simply saying, I know that there are trials for different resistant mechanisms. I have a G12C. This particular drug doesn't seem to be working anymore. I am curious to see if there are other studies. I don't think doctors would take that the wrong way. And I don't think you need to feel awkward about it. You're advocating for your care. You know, it's like somebody putting you on a blood pressure medicine and you get a headache every morning when you take it. You're going to go to your doctor and say, you know, this pill is giving me a headache. Do you have something else, right? You don't shy away from changing your blood pressure medicine because it doesn't agree with you. And this is as important or more important, right? Find me another trial, or let's biopsy and see if we can find something different.
Well, and it kind of does bring to the question is like, as far as clinical trials and second opinions, it kind of all falls in the same thing. Because not all places have the same clinical trials, right?
No, they don't. And that can be a little bit of a challenge. Um, and, and it does cause a little bit of a delay in care sometimes if a trial is not immediately available. Look, there are so many studies, there are so many compounds in clinical investigation. It's nearly impossible for one center to have all of the studies that are available out there. It's just not reasonable anymore, right? So, to some extent, the place that you choose to go to um, should have access at least to a network of other hospitals and centers where if they don't have a particular trial, at least within a reasonable driving distance, you can find a particular trial that might be relevant to you, right? But it is a problem we face in this country because it's such a huge country. And there are areas where you can drive for 300 miles and you don't hit a major cancer center. And, and, and that is not something that's fixable by us, right? We will never be like a small European country where everything is streamlined and you can take a train and 2 hours later you're someplace else where it just doesn't work for us like that, right? And the healthcare delivery in the US is kind of fragmented because the eastern parts of the country have different insurance companies, the western parts have a different kind of insurance company, the central, it's just completely different. And everything is sort of managed slightly differently. But the bottom line is that for things like KRAS, for these mutations that we identify in lung cancer or some of the other cancers right now, there are trials available that can be explored. And keep in mind that having a trial doesn't necessarily mean that things are going to be, working in your favor necessarily or anything like that. It just means there's a potential to have targeted therapy with the goal of specifically hitting the target that we're interested in. But at the end of the day, it's an experimental approach, so you could benefit from it, you could not benefit from it. So all of these things go into the calculation and makes it a little bit difficult to figure out what exactly is the next best option. But our goal, or at places like Fox Chase, is to say, look, what we have been doing for the past 20 years hasn't worked. These targeted therapies and immunotherapies are offering a lot of hope, and they work really well. Quality of life is better, responses are better, survivals are better. So we are on the right trajectory. We just have to keep the momentum going. And the momentum says, do another biopsy, find another resistant mechanism, do another trial, improve the science, improve the clinical care, move on to the next one. And that's what the goal is here.
So your goal is the same as ours, to get us healthy.
Absolutely. I mean, it's everybody's goal in this setting to sort of get to where we can actually say we are curing lung cancer. We realize that lung cancer comes in many, many different subtypes. But if you have paid attention in the last 10 or 15 years or 20 years, what have we done? We have identified specific mutations, alterations. We've made a drug. We've improved outcome in that particular area. We've tried to build on the knowledge that we've gained to make that particular mutation or subtype better. And then we have started looking for other mutations. So now we have drugs for 1% of patients with lung cancer. We have drugs for 22% of patients with lung cancer. We have drugs for patients with 2% of lung cancers, right? We are subdividing the lung cancer into smaller and smaller parts, but for each part, we're developing really good, effective, better targeted therapy, for the most part, pills. So the advancements allow me to say that if we continue this trajectory, if there's support, if there's funding to continue to look for more targets and more mutations and develop these drugs, we're going to get to a place where, yeah, you could be on a pill for a year and a half, and then another pill for another 2 years, and then another one for another 10 years, or so on and so forth. So that's the trajectory we're on. And my hope is that we won't lose this momentum. And my hope is that what we're learning with targeted therapies and immunotherapy and all that, we can continue to build on it and find better treatments down the road. But that momentum is hard to maintain. And it takes you guys to support and to be advocates for research and funding and all of that to continue this momentum the way we want to see it.
And we need to be the ones that, I mean, we stand to gain the most by doing the clinical trials. I mean, really. I mean, in the end, we're risking the most, but we stand to gain the most as well too, right?
Yeah, I mean, again, the bottom line is to make this disease, even if I can't cure it, if I can make it into, again, a blood pressure medicine that, you know, once in a while you have to change and go into something different. I think we have done a lot more in the last 10 or 15 years than we did in the past, in the 30 years before that in the world of lung cancer in general. And that improvement and all the data that we are seeing seem to suggest that, yeah, we're applying the knowledge that we're gaining appropriately. It's just that we don't want it slowed down. And that's the fear that I have, that all of a sudden we're going to slow this down, and then, you know, it's going to be another 15, 20 years before we have another major breakthrough, which is not something we want to see.
No, so we need to take this momentum and keep going with it.
Absolutely.
Because otherwise, I mean, you lose, it's like anything else. You lose the momentum, and then you lose having problems.
I mean, all this excitement, I mean, this wasn't possible. I never had an opportunity to sit in front of 50, 60 patients and caregivers to talk about one specific problem. And and I think that has been missing, right? I mean, that's the part that we love, and that's the part that we need, because without your support, whatever I do here, you know, might go to waste. There's just nothing I can do with it, right? And then you can be the advocate for more testing, more research, more funding, more clinical trials in a right way, right? And you can also be advocates for, well, my cancer has changed. Let's do another biopsy. Let's do another molecular test. And there's, again, these are the kind of things that you can push. And when you push for it, when you ask for it, then it becomes sort of the standard of care, because everybody out there says, you know, patients are demanding it. We can't say no to our patients. We have to do it. And that becomes part of the standard of care. And then everybody else can benefit from it too.
So it's okay to push.
Well, and I'm going to say something, hopefully it won't get me into trouble. And somebody told me if I ever say that, I should stop saying it. But why do you think the world of breast cancer is so much ahead of all the other malignancies? Because there was advocacy, because they pushed, because they did not accept the status quo when our mothers or sisters or wives were diagnosed with breast cancer. We were all up in arms. No one wants to see mom with breast cancer or wife with a breast cancer, right? We were all up in arms, and we still haven't stopped. Survivals with breast cancer is far better because there was advocacy and there was push. We need better treatments. That led to more funding, federal funding, philanthropic funding, walks, runs, cookouts, cookies, everything helped. And we got to a place where, the amount of money you spend on research and development correlates with how good survivals are in all of these diseases. For lung cancer, there was nothing. And one of the major issues there was no advocacy. But now there is, because it is a big problem, because we all realize what we've been doing has been inadequate, and because we are encouraged by the results of G12C stories, by the immunotherapy stories, by the EGFR stories. So the possibilities are there and we're all seeing it. So we're all excited about it, right? And we want to see more. So this is where you come into play. You have to advocate and you have to push.
We do. We do. And I personally, I think that organizations and groups like this coming together is really what it's about, and working across all the lung cancer organizations, because we all have the same common goal, right?
Yeah.
And do you, I don't know, we could get into a whole thing about stigma and all that goes along with it, but I do, my personal opinion on it that nobody cares about, but if it's about a biomarker, then it's about a biomarker, and really we don't even need to have a conversation about the stigma.
There are a bunch of questions in the chat. Are you looking at the chat?
Yeah, you want to grab one and go for it?
One is to put a shout out for NIH. It's, one of the jewels of our medical care here is NIH and the National Cancer Institute. Everything we can do to increase funding for the NCI and NIH, writing to our congressmen, to our senators, everything, I think we have to do it, because they do a fantastic job of funding critical research, funding clinical trials, genomic testing, everything. So all of us are, at awe of what the men and women who work at the NCI do, because it is really, really important. And, it's hard to do this. Other countries have tried to do this, but I don't think anybody has been that successful. So definitely visit the NIH website, the NCI website. A lot of good information about cancers, a lot of good information about clinical trial. There is a major database of every clinical trial in the country. So if you go to the website, you can actually put in where you live. I think it asks for a zip code, what kind of cancer you have and what stage it is. And you press a button and there's tens and hundreds of research and clinical trial options can come up for you depending on where you live. So that would be a really good source. And we are all obligated to register all of our clinical trials with the government. So it is all going to be in this database. If I am doing a study that I have designed at Fox Chase, I have to put it on this website. If Amgen or Mirati or Pfizer or Genentech, they're doing studies, they have to register the study. So this is a searchable thing, and it can actually help you find the right clinical trial within your area. So I think that's a really good thing to go to. Yeah, clinicaltrials.gov. Lynn just put that web address in there. So, and actually .gov, yeah. So visit that and bookmark it. This is a very good resource, and I would highly recommend it. The NCCN guidelines have been brought up. NCCN guidelines are people like us in academic centers. We get together. It's a guideline committee, basically trying to tell other physicians who might not be quite as involved in research and all of that, what are the best possible treatment options right now. They're guidelines, you can choose to follow or not, but because they're national guidelines written by experts who know the disease sites, they're highly valued. And it actually provides step-by-step evaluation, like what kind of tests you need for diagnosis, what do you do after diagnosis. So like for the lung cancer guidelines, it specifically says sequencing, next-gen sequencing for these mutations is an absolute necessary component of workup of a patient with metastatic lung cancer. So these are the kind of things that we go through. And NCCN guidelines almost like an algorithm, so it's really easy to follow. You go to step 1, and from there you go to step 1B, and then to 2, and then 2A. So it's simply laid out. So I think these are really, really good. There's a question from Wayne. Can you advise everyone here what's the best way to choose among all the different trials available out there? That again becomes a little bit difficult because first of all, you have to be able to get to a center. So, can you get to the place that does it? If you have multiple options, which one it is that makes a lot more sense to you? Let's say I can explain the trial, but if it doesn't fit right with you, then maybe that's not the right fit for you, right? And then there are clinical trials that ask you to have 3 biopsies. Well, you might not want to do 3 biopsies and choose something that has 1 or 2. The problem here is that when a new drug comes and it's really hot and it looks good, the hype is out there, so you know about it and you can look for it. But when we come up with a new drug and there is a new clinical trial, it is still an investigation. So, in good faith, I can't tell you this trial is definitely better than the other one. I can tell you what I think about the studies, and I can tell you one is a little bit easier, the other one is a little bit more complicated. One needs you to be here 5 days a week, the other one needs you to be here, once every 3 weeks. So those kinds of stuff I can tell you, but like from an ethical point of view, I cannot tell you that Hossein Borghaei's trial is better than Terri's study because Hossein Borghaei did it and Terri didn't, right? So that becomes a little bit more difficult. Those are the kind of measures that you have to use to sort of figure out what trial is best for you, and that's I think it's a little bit more difficult for me. Tessa says, I agree, need to keep this momentum going, really push. I agree with you. Imagine put the fraction of the money into cancer research that has gone into COVID. Yeah, but at the end of the day, COVID is a global problem that's bringing the entire world to its knees, right? The economic impact of COVID, even if you look into your own neighborhoods and cities and all that, is devastating. It's really hard to see people being homeless and out of jobs and things like that because of a pandemic, which I never thought we would see it in this country, but here it is, right? We all have to deal with it. So I think the reason all the money went into COVID is because the world was shutting down, and you sort of kind of understand that. But you're right. Now that COVID is behind us, maybe we can ask the president to put a little bit more money into NCI. Now he already has allocated a lot of money, and that he has an interest in cancer and all of that, but we need to tell our elected officials that this stuff is important to us. So maybe a little bit more money for the NIH and NCI would be a good thing. And again, that requires advocacy. You gotta talk to politicians. They're the ones who decide who gets the money and how to spend it. So the more they hear from us, the more they understand that these things are important for us. So absolutely. What else do we have?
How necessary is a repeat biopsy if you've already had full genomic testing and experienced progression on a non-mutation specific inhibitor?
Good question. So no, you're right about that. The rebiopsy and resequencing makes sense if you have a targetable mutation, like the KRAS G12C, EGFR, ALK, RED, those kinds of stuff. That's where we are looking for these bypass resistant mechanisms. If you had testing at the time of initial diagnosis and you did not have one of these alterations and you went on chemoimmunotherapy or immunotherapy alone, right now there is not a major push to do repeat biopsies because you're right, there's not a lot we can do with it. Now, again, at Fox Chase, we have one protocol where we ask for a repeat biopsy if you're on immunotherapy and cancer is not responding because we want to know why that tumor is not. But that's more of a biological endpoint trial, not the drug trial. So you're absolutely right. If you don't have a targetable alteration at the time of progression, there's really not a lot of need for repeat biopsy. So that's a good question, and thanks for asking it so that I can clarify it. Why is genetic testing limited to advanced metastatic disease and not first surgery? Well, that's changing. It's not for KRAS yet. Hopefully we will get there because there are studies looking at it. The reason it is for the metastatic disease is because a couple of things. A, we develop drugs first for the metastatic setting because that's the incurable state where we need better treatment options. If they look good and promising and safe, then we feel more comfortable taking them to the early diagnosis. Why? Because when you're diagnosed with an early stage disease, your chances of cure are much higher. We don't want to bring a drug in there and make things worse. So If it works in the metastatic setting, it is not toxic, it's really good, then we can say, all right, what happens if we bring it to an earlier stage? Can we increase the cure rate in an earlier stage? Example here is the EGFR mutated lung cancer. You've heard about EGFR just in the context of metastatic disease up until 6 months ago, where all of a sudden the same EGFR drug that was being used in metastatic setting was used in patients after surgery, and there was a phenomenal response, a delay in re-emergence of drug. So the drug was proven to be safe, effective, tolerable in the metastatic setting. Only then we moved it into the curable state, and that data has now been incorporated into the NCCN. So these things take time. We don't want to mess with the curative patient population if the drug is too toxic. You can imagine that doesn't do anybody good. So it takes time to move them into the front line. Immunotherapy, everything was in the metastatic setting, absolutely everything. 2 weeks ago, or last week at AACR, one paper presented, now immunotherapy in the front line could, the data is not complete, but could be used and might be good, might actually be able to cure more patients, but the data is early. So now we know immunotherapy is okay, it's safe, we can move it to the front line and now, or to the earlier stage. So we're doing those studies. So that's the way the drug development always works in oncology. So it just takes time, unfortunately. KRAS amplification, are there trials? I am not aware of anything for KRAS amplification at this point, so I'm sorry. I can look that up and communicate that with Terri and get back to you on that, but I don't know of any right now. If you have a recurrence, you may not be able to have tissue taken for sequencing. That's true. I'm asking about genome testing and when it is done. If not done with tissue available, more tissue may not be accessible. So this is a common problem in the world of lung cancer. Sometimes we just can't get enough because the tumor is sitting in a really funny place. You can't put a bigger size tumor because it could be dangerous and you get a small piece. There are blood-based genomic assays, 2 tubes of blood drawn in the clinic, sent to these companies that do this, and you can potentially get your mutational panel just from 2 tubes of blood. The available data says these blood-based platforms are very, very good if they detect a mutation, but there is a 20% chance that they can't detect anything. In that case, you still have to go back and do another biopsy if feasible. So the blood-based assays are becoming very popular. They are very useful. They do seem to be accurate, and they could, in these situations, completely replace tissue biopsy. But it's not complete replacement. The data sort of suggests the best thing to do is to do the tumor and the blood together, because then you can find even more mutations. But as I said, that just sometimes is not possible, and you can get more tissue. So that's why the blood-based assays are helpful and can be very useful in this setting. The dogma or the teaching is, if you only do blood-based assay and the blood test is completely negative, you must do a tissue biopsy and send it for sequencing. But if you do a blood-based and you do find a mutation, then you're done. You have your mutation and you can be confident and you can act on that. Does that make sense?
Is there ever a chance of erroneous results?
You mean false positives? That's very rare. But the negative blood, the blood test happens more often than the negative tissue results. Does that make sense? That's why when you have a completely negative blood assay, we recommend that you consider another biopsy, get more tissue, and do the tissue biopsy. Okay? Is it important to know whether you are sensitive to chemo? Many people start an IO and then move to a trial without trying chemo. There is really no good way of testing the tumor to see if it's sensitive to chemo. Now, there are businesses that sort of say they can do that, but it really doesn't work like that. So there's really no good way of doing that. I personally think that chemo still is effective. Now, I'm going to explain to you there's a little bit of a generational gap now, and I'm right at the generational gap in terms of doctors taking care of lung cancer patients, where doctors older than me believe in chemo 100%. I believe in chemo and want to get rid of it because I think chemo has a lot of side effects. And then there are the people I am training now. I'm training them to think of a day where chemo is something that we lose all the way down the road if we have to, kind of a thing, right? So there's a little bit of that. But in lung cancer, chemotherapy is effective. You routinely get a 20-25% response rate, meaning some shrinkage out of it. So do I support the fact that somebody just goes on IO and never wants to be on chemo? I can understand it if you tell me I absolutely do not want chemo, and I'll do my best to treat you without chemo. But we have to realize that even immunotherapy doesn't work in everybody. And if it doesn't work, the chemotherapy option at least gives us that 20-30% response rate and all of that. That's good. The data with chemo plus immunotherapy is really good, but we are seeing more and more patients coming to our offices and saying, look, whatever you do, I'm fine with it, just don't give me chemotherapy. And now we have options. We can actually work with that. But in some cases, I'm not sure I can advocate for it. And it's my job to tell you, yeah, you can tell me you hate chemo and I believe you and I want to try it without it. But if I really believe that it can help you, we're going to have that conversation and then we're going to have to decide how to proceed. Okay? How well do KRAS drugs work across cancer types besides lung? We're trying to figure that out. The data I have seen so far says the currently available G12C drugs work much better in lung than they do in colon cancer. Why that is, I cannot tell you. You need to talk to a real biologist who sort of looks at this in the lab. But right now, I cannot explain that. Now, the work isn't done. The clinical investigation in the colon with the KRAS drugs is continuing. But the initial report that I have seen says that it works better in lung for whatever reason. So we have to wait and see. Is KRAS G12C responsive to chemo? Yeah, it's also, if it doesn't have a lot of other comutations, it's also responsive to immunotherapy. So there are a lot of good options for the treatment right now. I'm sorry, immunotherapy didn't work for G12V and caused more problems. Yeah, I mean, unfortunately, these things can happen. I mean, these are really powerful drugs. And we go by, you know, the available data from clinical trials. And what we see quite consistently is that not all of our drugs work in 100% of our patients 100% of the time. And that's just the nature of this disease, unfortunately. And again, another reason why it is so important to support research and better understanding of all of this, because yeah, unfortunately, even the best drugs that we have sometimes doesn't work in a patient, and sometimes it can cause more problems than the hope of, you know, activity and control. So I'm sorry to hear that, but unfortunately, these things can happen. Nano chemo. I think it refers to the use of the nanotechnology, these particles that sometimes are coated with gold, sometimes with other things that can deliver high levels of chemo or other drugs specifically to the tumor site. I think that's what you're referring to, the nano chemo, use of nanotechnology to deliver chemo specifically to tumor. We've been playing around with this for quite some time. Again, another area where the technology is really getting interesting and getting really good, but not ready for prime time as far as I'm concerned, under investigation. So we'll see. And then somebody's trying to get me into trouble. In your, which is more effective, Amgen versus Mirati trial for G12C? I don't know. We haven't had. So what the question is that there are right now 2 companies that are in the lead for development of specific KRAS G12C drugs. One is Amgen, the other one is Mirati. Just so that we're clear, I work with both of these companies. I've been consulting for both of these companies and I have their trials. So I'm involved with both of them. I cannot tell you which one is necessarily better because patient populations are different. And there is no head-to-head comparison, and more data is needed to really see if one is necessarily better than the other one. So I won't be able to answer that question. I'm sorry. But what I'm happy about is that at least we have two options and hopefully more to come. More options, the better, right? And remember how we had only one Pfizer vaccine for COVID, but now we have Mirati, and then we have another one, and have another one. More option, better for all of us. Thank you. Oh, you're more than welcome. It's a pleasure being here and talking to you guys, of course. Can T-cell therapy work for G12V for KRAS? Depends on what you call by T-cell therapy. If you're talking something about like a CAR T, where you make specific CAR T against G12T and all of that, I would say, yeah, it is a possibility that it can work. But the studies have to get done. You have to make the CAR T cells and you have to go after it. There are different kinds of T-cell therapies or cellular therapies now that they don't necessarily involve the manufacturing of CAR T cells, which are a little bit more sophisticated and more complicated and all that. So the T cells that can go inside a tumor, that's when we think the tumor is a response. So if I biopsy somebody who's responding to immunotherapy and somebody who's not, more likely we can find these T cells in the tumor of the person who's responding to immunotherapy as opposed to the one who's not. So a bunch of really smart investigators say, well, what happens if I take these T cells and give them to patients who are not responding? Can they find their way inside the tumor? And those are the kind of experiments or the kind of clinical trials and all that that are ongoing. So the possibilities are there. It's just that we don't have a lot of data yet in support of it, but stay tuned. I think you're going to see some very interesting studies coming up. Another good question. If sotorasib stops, that's the Amgen drug, will adagrasib, which is the Mirati drug, work as an option? I would say probably not, but that has not been tested. So I'm not sure I can tell you that that's going to be a good strategy going forward. If we make specific drugs to whatever happens after either sotorasib or adagrasib stops working, then that might work. But right now, I think these drugs are very similar to each other, so I'm not sure that's necessarily going to work. Anything with cutting edge drugs data coming out for lung in general that isn't KRAS specific, chemoimmuno that can be used to fight KRAS. Well, chemoimmuno can be used to fight KRAS. The reason we like targeted drugs is what Terri referred to in the beginning. I like to take a pill and not have to worry about it, right? Chemoimmunotherapy requires IV administration of drugs. You come to the hospital, you get to a cancer center every 3 weeks. You have to do this, you have to do that. But yeah, chemoimmunotherapy by all accounts can work. The refinement here is that, remember how we were talking about comutations a little while ago, about half an hour ago? Mm-hmm. Some of these comutations could make chemoimmunotherapy less effective. So that's what we're trying to figure out now. So it's not just the KRAS G12C or G12V. Is there something else there that can make the chemoimmunotherapy a little bit less effective? And if that's the case, what do we do about it? So that's the question that we have right now. Hey, I'd like to thank all of you for being here and giving me this opportunity to talk to you. I'm sorry if I talk too much.
Oh my gosh, no, this is great. This is fabulous. This is fabulous. Okay, well, I can see Jackie like followed up. I mean, is there anything else? I think she's looking to see, is there anything cutting edge that's going to help KRAS that's not like specific, like sotorasib or adagrasib?
It's more general, like the T-cell therapy. I think that is something that's more cutting edge and probably coming to a lot of the offices and hospitals and cancer centers soon. I really do think these cellular therapies have a lot of promise. The CAR T cells, I think, can carry the promise. It's just that they're complicated. They're not easy to apply in every setting, but they can open up the way or the door to yet other forms of treatments. I think these are really cutting edge technologies that we're trying to utilize. Cancer vaccines are becoming very popular. Now, these are not your grandpa's cancer vaccines that never really worked, unfortunately. These are really sophisticated cancer vaccines that are beginning to show some promising activity early on. And, I'm excited about it because I kind of like immunotherapy and vaccines and all that. But I think these very specific, lung cancer vaccines, or better yet, patient specific vaccines, could have a lot of utility in the management of the disease. It's just that the research is early on, and we're going to have to sort of identify ways to make it applicable to a wider patient population. So there's a lot of really good research going on. New immunotherapies are coming out, and some of those can be very effective. Forget about what you've heard. I mean, there are other immunotherapy drugs that are being investigated that could potentially be very, very active, either by themselves or in combination with other currently available immunotherapy drugs. So there's a lot of research that's going on that I would consider cutting edge. Hopefully, we'll get to use them soon and have a lot of benefit for everybody involved.
Well, I don't want to cut you off. I'll keep you here all night, and we'll keep pelting you with questions. But I do appreciate your being here, and I want to be respectful of your time too. And so you've made this like crazy easy for me. So, so I really do appreciate it. And I'm going to ask everybody if they want to stick around, we'll do a photo, but only if you want to be in a photo, please stick around. We'll do a quick photo. But I do want to just be so ever so thankful, truly, for having you come on and being so open and so, accessible to be able to answer our questions.
Mean this seriously. I really look forward to this. This is a great opportunity for me to talk to all of you, hear some of your concerns and some of your questions, share it with my colleagues, and hopefully build on this. So Terri, again, thank you for giving me the opportunity. I'm looking forward to working with you even more on these sorts of events and others.
Yeah, this is going to be great. Okay, well, if I can get you, you need to stay because you're a little centerpiece here. So if y'all are okay